Welcome to the George Miller talk and to Lynn Nadel, who will be giving it. Hi, everyone. It's a joy and an honor to introduce Lynn Nadel for this George Miller lecture. So let me begin by saying Lynn and I go back a very long way. After ascertaining that his kids aren't here, I think I know him longer than anybody else in this room. I was an undergraduate at McGill from 1962 to 66, and Lynn became a graduate student in 64 working with Dalbir Bindra. It was just a wonderful place to be. The atmosphere at McGill, aside from an interest in memory, the graduate students and undergraduates all formed one group, and I remember many of us just staying there till midnight and then going out for some food and then coming back and continuing to discuss research. Actually, it's a little crazy now that I'm saying it. I should tell you, I've asked the organizers if Lynn and I could switch. I have 5 minutes for an introduction, and Lynn was given most of the time for a talk. But I've known him for such a long time, I thought I would launch into everything. I'm sure Lynn wouldn't mind. So let me tell you just a couple of things about Luck and Lynn Nadel. It's an interesting story. After when he graduated in 67, he got his Ph.D. in 3 years, which I took as my benchmark, thinking that everybody gets their Ph.D. in 3 years, but that wasn't true. Not for me and almost not for anybody else I know, except Barry Schwartz, actually. He went to work in Prague with Jan Buriš and Olga Burešová to study memory and perception, I think, and what is it? Spreading depression, I think, is what they were known for. So the only good thing that came of Russia's invasion of Prague, of Czechoslovakia, and the end to the Prague Spring and to communism with a human face was that it forced Lynn to leave Prague for at least a little while and begin a collaboration with John O'Keefe. So we're all beneficiaries of Russia's invasion. Sometimes good things happen, bad things turn out to have at least a bit of a silver lining. Now, we all know Lynn for his memory research, but let me just mention a few things before I get on to the rest of the introduction. Lynn does basic research on the neuromechanisms of memory and developed the cognitive map theory with John O'Keefe during that time that he returned to London. In addition, he's done work on memory consolidation and reconsolidation. There's also a clinical aspect to Lynn's work. He's done some very important work on memory and clinical syndromes, especially Down syndrome, for which I believe he was honored with an award by the Down Syndrome Society, if that's what it's called. And he also does work on the role of stress and memory, and that he's now started to apply to developmental disorders and also to try to understand some mood disorders and anxiety disorders. So this isn't the first honor that Lynn has received. So in 2006, he won the Grawemeyer Prize for Psychology. In 2020, he won the APA Lifetime Achievement Award, and he was elected to the National Academy of Sciences in 2022, I think it was, or is it 2021? I can't remember. It doesn't matter. He's in the National Academy. Nobody looks at the date. So if any of you really want to know something about Lynn's trajectory, there's a wonderful profile in PNAS, in the April 2022 issue, that describes Lynn's trajectory in science, and also there's also a paper that bears the same title as this talk today. So if any of you want to go to sleep now, it's a good time. You can always read it in PNAS. I'll tell you just a couple of more things then before I let you go on. So you can read about his professional life, but let me tell you a little bit about his personal life or interests. He's interested in very, very many things. He's an avid sports fan and athlete, but in terms of sports fans, it's just New York teams, as far as I know, that he likes. But he follows everyone. He likes games. When I was an undergraduate, Lynn taught me how to play 30-second Scrabble. So we'd go and run our rats. I did rat work at that time. And when there was a break, we'd come into Lynn's office and play 30-second Scrabble with a chess clock. He was a wonderful, I don't think I won one game. Lynn is married to Mary Peterson, who's here today, and also, I don't know if she will be speaking, but the best thing that I want to tell you is the role of luck. I mentioned it already about being forced out of Prague, but the other thing that I want to mention is that Lynn's cognitive map theory, if I understand it correctly, probably not all of it, Lynn always wanted to focus on the spatial component, but there is another component to it, which is to be able to create a very rich cognitive representation that allows you to move from one part of that representation to another. We can see this in Lynn's research. He manages to link together work that other people couldn't find a way around the maze, really, to begin with, but he already saw these connections. And he's a wonderful writer. Having written with him, I think I wouldn't have gotten nearly the credit that I did if Lynn hadn't been around. He has a clear idea of the goal and knows how to take shortcuts to the goal. I tend to be a kind of person who likes to wander around and look at the scenery, but Lynn actually doesn't. He likes to get right to the point, and it does it very, very well. All right, so now for the citation. Let me see if I have it here. To let you know what he's getting the award for, if any of you have not yet guessed, let me tell you what the criteria are. The 2024 George Miller Prize goes to Professor Lynn Nadel, whose career is characterized by a distinguished and sustained scholarship and research at the cutting edge of our understanding of nature of memory processes observed by the hippocampus and medial temporal lobe. I mentioned some of the other things that he's done, but I can't find, sorry, I had the George Miller citation. In any case, what it says is that it's for a lifetime of creativity that brought us to the cutting edge of research and managed to push our field forward. So he's done it in both basic science, and for those of you who are probably less familiar with it, in clinical science, I would say. So without further ado, Lynn, I think you're free to talk. I'm done. Here's the George Miller Award. And here, this is the other stuff that you can keep later. Okay. Thank you.
All right. So what Morris didn't tell you was that I was the TA in a course that Peter Milner taught, the other Milner, fourth-year course in physiological psychology, and Morris was a student in the course. And he, of course, was the best student in the course. And that was where our friendship started, and it hasn't ended. That was 1964, 65, something like that. So it's an honor to be introduced by Morris Moscovich, let me tell you that. All right. First of all, I want to thank the Society and the selection committee for choosing me. It's a great honor to be linked with George Miller. I don't know how many of you, can you all hear me? Okay. I don't know how many of you know about George Miller. You should, of course, 7 plus or minus 2 and so on, but there was more to George Miller than that. He wrote a book in 1960, co-authored a book called Plans in the Structure of Behavior, which all of us who were graduate students in the 1960s read from start to finish, because it was one of the earliest books that actually influenced the field in a very strong way. So George Miller, to be sort of in the same sentence with George Miller is quite an honor, and for that I thank this society. You see that he felt he was persecuted by an integer. I've been persecuted by many things, but not an integer. All right, so my approach is that a good science talk is kind of like telling a story, basically, and getting a narrative, that sticks in people's minds. But there are stories and there are stories, right? So there's this story, the Ted Lasso story about, be like a goldfish. They're really happy because they only have a 10-second memory. That's, of course, a false fact. That is not true. I'm going to come back to this right at the end. Then there are weird stories. Here's a weird story. Quantum entanglement. I don't know how many of you have ever heard this term or know what it's about. But it says right up there, I'll read it to you. When two particles, such a pair of electrons, become entangled, they remain causally connected even if they're at great distances from one another. It's a rather spooky thing, so to speak. And that's what Einstein called it. He said this is spooky action at a distance. And other physicists said that physics should represent reality in time or space without spooky action at a distance. So that's the way physics feels about action at a distance. Trouble is, we're not physicists, most of us. Some of us may have been trained as physicists, but we're mostly not. We're behavioral and cognitive neurobiologists, and what we study is not reducible simply to physics, because unlike physicists, we are always studying behaviors that routinely involve action at a distance in one way or another. So behavioral adaptation involves action at a spatial distance based on things in places we can't directly sense at the moment. And it also involves behavior based on past events or thinking about behaving with respect to something in the future. So in some sense, we're always behaving at a distance. So in nature, this is nature. This is a picture of the Serengeti plain. I take credit for this picture. It's an amazing place. Go if you get a chance. But in nature, life's essentials are often at a distance. They're beyond the reach of one's senses. So animals navigate efficiently by using previously acquired information, where they are in the world, where critical resources are located, how to get from one place to another, and so on and so forth. So the proposal I'm going to defend in this talk is the notion that the hippocampal formation creates representations of context, cognitive maps, that enable organisms to bridge spatial and temporal gaps. That's the basic claim. Now, this is not an entirely new thought. In fact, it's been speculated before. So here are a couple of quotes from fairly old papers. Nick Rawlins, back in 1985, said something about the hippocampus being needed to associate stimuli that are temporally discontiguous. Wallenstein, Eichenbaum, and Hasselmo said something quite similar. The hippocampus is necessary for associating discontiguous events. So the thing is, neither of them followed this idea up in a systematic way. They said it and moved on to other things. I'm going to explore that idea in some depth in this talk, and I want to make the point that we bridge these gaps, spatial and temporal gaps, at multiple scales. Here are a variety of ways in which we bridge this gap, phenomena that involve bridging this gap. We migrate across vast spaces, we navigate to things we can't see, we identify contexts, we map contexts, and so on. I'm not going to talk about all of these things. I don't have enough time. But I am going to talk about the ones that I bolded here. I'm going to talk about mapping and identifying contexts, about learning contingencies, and about indexing details. And then I'm going to say something about memory updating at the end. All right, let's start with map construction. So the thing to think about, and I have to admit that I didn't think about this when we wrote the book way back when, is that map construction occurs piecemeal, basically. Over multiple separate visits, animals and humans develop increasingly rich internal models of an environment, updating it as is required. And in fact, we've been hearing in some of the talks that were just in the session before about this need to integrate information across multiple experiences. But here's the key point here, that this piecemeal construction is not actually straightforward. It doesn't just happen kind of by magic. It requires a critical step. And the critical step to form good memories, that in fact bridge time in that way, is that information about the same context gathered on separate occasions has to be linked together. And this doesn't happen automatically. The brain has to retrieve the appropriate internal model, its context representation, upon revisiting a familiar context if it's going to update information about that context. So that's the critical first step, which is re-identifying the context so that you can aggregate information across multiple exposures to the same context. So the first key question is, where am I, basically? And it's interesting to note, by the way, that not knowing where you are induces incredible panic. This is something that we're not comfortable with, not knowing where we are. There are many things we can be comfortable with, but not knowing where you are is not one of them. So that could be, if you know John O'Keefe, that could be him, looking, but that's what he looks like from the front. I'm not sure I don't think that picture is of John. Anyway, the point I want to make is that the brain connects information gathered on sort of different occasions, different visits to the same location, but only after recognizing or re-identifying the context, using stable landmarks. This figure is from a computer model that is built to recognize contexts, and the way it works is by picking out stable landmarks that it can re-identify, and then it says, oh, I'm in this particular context. So that's the way, and animals know they're in the same place by re-identifying the context in this way. So, just to put it to you bluntly, most of you probably hadn't thought about the fact that re-identifying context is actually fundamental to how and what animals learn about the world and how they act in it. Here's a quote from a very recent paper. I'll read it. "The computational task of inferring the current context is non-trivial, and the way the brain solves this task has profound consequences on the processing of memory." Okay, so let's talk about learning contingencies. So here's a very basic question that psychologists have, not just psychologists, Aristotle also wondered. What leads to learning, right? There are two kind of different perspectives, you might say, amongst, I'm simplifying, obviously. One idea is that contiguity leads to learning. This is the standard idea, standard association theory. Stimuli that are close to one another in time and space become associated, so proximity is crucial. That's one notion. And it leads to these kinds of classical conditioning paradigms that you're all familiar with. The other idea, which has gained much currency, is that contingency is what leads to learning. Stimuli that depend upon one another become associated. Predictive value is crucial, not contiguity. And that leads to different ways of thinking about learning from a modeling perspective. Of course, many of you know Rescorla and Wagner, now this has got to be like 50 plus years ago, pointed out that really learning is mostly driven by contingency, not by contiguity. Now, temporal contiguity can be, and usually is, established by a single experience. Two things happen together once, and you think, wow, they must be connected. There lies the path to conspiracy theory, by the way. Contingencies, on the other hand, are probability estimates. They require aggregating information across multiple experiences so you can actually determine, compared to the base rate, whether something really is predictive of something else, right? So it's a different story. And extracting contingencies typically requires bridging temporal gaps in most of the experiments we do. Because in our experiments, our rats or our mice or our humans, they don't stay in the experimental apparatus for the whole time of the experiment, unless it's a one-day experiment. They go home. They go to their home cage. They get taken back to their home cage. The next day, they come back out of the home cage, and they're put back in the apparatus, and you don't pay attention mostly to the fact they have to remember, the context. They have to remember what it is they learned in this situation before. It's not something that just happens magically. Why does this matter? Do you think this is trivial? I'm going to give you an example of why it matters. This is a very famous figure in the history of what has become cognitive neuroscience. This is the paper that Stu Zola Morgan and Larry Squire and Alvarez, whose first name I can't remember, I fear, published in Science in maybe the mid-1990s to demonstrate that the hippocampus, the medial temporal lobe in the hippocampus was a memory system, because in this delayed non-matching to task, performance of the monkeys with hippocampal damage fell off when the delays were extended to 10 or 40 minutes. That's the finding, and it was the backbone of that theory. Unfortunately, what was not pointed out in the paper but became clear at one point was that the monkeys in the 10 and 40-minute condition, they were taken out of the apparatus and brought back to their home cage, so that when they came back to make their choice, not only did they have to remember which one to choose, they also had to remember that I'm in a certain context where that matters. And if I'm right, their problem was not that they couldn't remember anything, but they had no idea of the context that they were in, given their damage. All right, so basic story here. The brain uses spatial context representations to re-identify environments, to integrate information gathered over multiple visits to an environment separated in time. And this allows them to map space across time. And these are mapped at multiple scales. But first, a bit of a sidebar. If the hippocampus is indeed, as I'm claiming, crucial to bridging temporal gaps, and if bridging such gaps is crucial to calculating contingencies, then hippocampal damage should disrupt the learning of contingencies. Straightforward, right? Turns out that in 1980, this experiment was done, so to speak. Nobody pays any attention to this. I paid attention to it because the L in Davenport, his first name was Lynn, the same as my first name. So there were so few people with that name that I automatically paid attention to anybody who did work who had that first name. He was the first one I met. Anyway, he did this experiment, and I highlighted in red the conclusions. You can read for yourself. Superstitious operant behavior characterized the behavior of rats with bilateral hippocampal lesions. And he then proposed the hippocampus permits the control of behavior by contingency. And without the structure, behavior is guided by simple response reinforcer contiguity. He had it all right there, right? And nobody paid the least bit of attention to it, except for me. But even then, I didn't understand why it was so important. All right, let's talk about how organisms identify contexts. But here's the elephant in the room. Right? Another picture from Africa. What is context? Right? Not a simple question. Broadly speaking, how I'm using the term, it refers to the background stimuli that accompany some kind of foreground event. Right? It's the kind of stable stuff within which dynamic things happened. That's kind of the technical definition of context at some level. But one can have a rather broad definition of context. One can say temporal context, emotional. I'm talking about spatial context. That's my focus. And spatial context can be defined as settings within you experience certain events, and you can return to them in reality or in your mind. You can think about them as well. And we build these internal models of spatial context or cognitive maps based on what I've already said. So here's a spatial setting that is meaningful to me. This is Wenceslas Square in the center of Prague. This was, you see 3 pictures. I was pretty much there in all of these occasions. The first one is during the invasion that Morris referred to. The second was during the kind of fall of communism in 1989. And this is what it looks like now. I don't know how many of you have been to Prague. It's a fabulous city. But to me, this is a spatial context I return to both physically and in my mind on frequent occasions. That's a context. But contexts can be either large, like that, or they can be small, like the computer background in an experiment. And that's really most of the kinds of contexts that we use in our experiments, or something that we impose on the stimuli that we use. But in either case, context is frequently ignored. Most people don't measure it. They don't measure its importance. They don't look at it, although it appears to be a part of what's learned, whether it's attended or not. So now I'm actually going to get to a little bit of data. And the data I'm going to show you kind of come from various points in my career, so to speak. So here's an experiment that was done with some colleagues in Arizona, Lee Ryan and Devin Duke and Kevin Chun-Yu Lin and I can't remember, Kevin Kawa. We presented them with pictures of objects on tables, basically, in various combinations. And then we tested recognition by either presenting them with the identical object in the same context or a familiar object in a different but familiar context, so familiarity is just the combination that wasn't the same, or by a new object in a new context, or on no context whatsoever. In this experiment, the participants were told, the background doesn't matter, just remember the object. That's what you're going to be asked to remember. And what we found across all of these conditions is that as soon as you move out of the identical object in the identical context, there's a fall off in recognition between 10 and 15%, no matter what condition you choose. So the absence of the background context, even though it's not part of the experiment, has caused a fall off in performance. We then did it we did the experiment again in a couple of different ways, using incidental encoding, intentionally. We told them what we were doing. We blocked the types of trials. It didn't make any difference. What you're seeing now on the, I guess I have a thing here. This is how much fall off there was in each of these conditions. There was always a fall off, and this was what we called the context shift decrement, so to speak. So being tested in a different context, even if the context wasn't part of what you were being asked to learn, affects your performance. So now the question then became, is the context involved in the creation of these kinds of context representations? So now I'll take you back, way back, to the dissertation that Morris said I got done in 3 years. I was 7 at the time, I think. So how does a context, here's what, in this experiment, rats were put in a box with a water tube, and they were trained to lick from the spout. This was a classic experiment back in the 1960s. It's called the conditioned emotional response experiment. That term isn't used anymore, CER. Having trained the animal to drink from this water spout in context A, I then put the animal in context B, which essentially was the same kind of box, but it had grid floors, and I sounded a tone and gave them a shock. This is classical context fear conditioning that people use now all the time. It wasn't called that then. And then on day 3, I think it was, came back, put them in context A, back with the water spout, no grid floor, the different context, and in a 3-minute session, in the middle of the session, I sounded the tone. And now the question was, how much fear would the animal show to this tone in this situation? I was not interested in the context shift. That was not part of my thinking at the time. And what I found, quite curiously, was that the rats with hippocampal damage, that's these guys here, the orange bar and the gray bar, those rats showed more fear in this context than the control rats. At the time, I had no idea what this meant. So I wrote it up. It was part of my dissertation. I got my Ph.D. I went on my way. There's a lesson in that, I'm sure. But years later, I realized that these data implicate the hippocampus in context coding, and that in the absence of a context representation, animals with hippocampal damage will generalize fear from one context to another, more so than animals that are intact. In some preliminary, this is a very old slide, in some preliminary data, we more or less thought we demonstrated that the hippocampus was not just representing contexts, but it was representing specific contexts. So this subtraction sort of separates out, when they're in the identical context versus when they're in familiar but different contexts. And there's a little bit of, you can see this bit of sort of activation in what looked to be the anterior part of the hippocampus. But again, preliminary data. We went on to do another experiment, and here one of the authors of this experiment, Jess Payne, is sitting right here. Another one of the authors, Siobhan Hoshite, was in the room when I gave this talk back in Arizona a few weeks ago, and this is Lee Ryan. In this experiment, we gave the participants sort of an interview about events that had happened in their life, and we asked them to recall various spatial and non-spatial aspects of these things, and we used that to create questions, which we then asked them in the scanner, basically. There were true-false questions, and they were of various types. They required the retrieval of various kinds of information, spatial episodic, non-spatial episodic, and so on. We got, this is not what I want to talk about now so much, but just to show you, we got what we expected to see, a main effect of space. The hippocampus and parahippocampal gyrus were more interested in spatial stuff than non-spatial stuff, and more interested in episodic than semantic. That's not the focus of this talk. This is the part that I'm interested in. We took the spatial questions, and we separated them out into questions that were relational, about, such as, at the wedding, your sister sat to the right of your mother. They were very specific spatial relations, versus locational, where the wedding took place in Aruba, kind of this context. And then we looked at the data from those questions separately, and what we saw very clearly was that the questions that were about location activated more the anterior hippocampus, and the questions that were about relations activated posterior hippocampus. So there's a clear difference along the axis in terms of what kinds of questions they sort of were interested in. Relations, specific relations in the posterior hippocampus, the dorsal hippocampus, and context or locations in the anterior hippocampus. So now, to go back to sort of a historical fact, you probably know that if you look at where the hippocampus is located in humans and rats, it's rather different. The vast expansion of the cortex in humans pushed the hippocampus down into the medial temporal lobe. In rats, there's still a lot of hippocampus up at the top. And just because it was easier to get there, the dorsal hippocampus, and this still remains true, by the way, the dorsal hippocampus is what most rat people and mice people work with, because you can get there more easily. The ventral is buried deep in the brain, harder to get there, so to speak. In humans, and HM in particular, his surgical lesion, was in the anterior hippocampus, right? Because the surgeons came in from that side. So this difference was actually what propelled my Ph.D. dissertation, where I compared dorsal and ventral hippocampal lesions. I had the crazy idea that maybe the disjunct between the animal and the human results from hippocampal damage, and there was a disjunct at that time. There was a memory deficit in humans, and there was no comparable memory deficit in the rat work or the monkey work at that time. This was in the 1960s. I thought, well, they're looking at a different part of the hippocampus. I'm going to prove, that they're both right, and I'm going to show that the dorsal hippocampal lesions will replicate what you see in rats, and the ventral hippocampal lesions will replicate what you see in humans. Again, that didn't work out, but it is an important fact of the matter. This long axis of the hippocampus has a really fascinating property. Space is mapped at ever-increasing scales as one moves along the axis. So here's data from I think this was the first experiment that showed this, done by Bruce McNaughton and Min Jung and Sid Wiener, who's in Paris. This was done in Arizona in the 90s. And this shows you that the size of the place fields in the dorsal hippocampus is smaller than the size of the place fields in the ventral hippocampus. That's a changing scale, and this has been developed now everyone agrees that this is the case. The same thing is true in the entorhinal cortex. How many of you do not know what a grid cell is? Good, everybody knows about grid cells. The grid cells in the medial entorhinal cortex show exactly the same granularity change along this axis. The sort of granularity of the grid shifts from being tight at the dorsal end to broad at the ventral end. Well, that's kind of interesting, but what's particularly interesting about this is that when you expose the animal to an alteration in the environment, the way these cells in this sort of ensemble of cells responds to this manipulation of the environment differs along the axis. At the dorsal end, you can see this, see if I can show you how. What you can see at the dorsal end is that only a portion of the representation is maintained at the dorsal end, but it maintains the metric quality of the representation. At the ventral end, the whole representation is maintained. It loses the metric information, but it keeps the information about the whole context. So these two parts of the hippocampus, which presumably are doing something similar computationally, are coming to different conclusions because of some of the way they're wired up. One end maintains metric information and detailed local information, which is why the dorsal and posterior hippocampi are crucial for navigating in space, and the other end preserves information about the context as a whole. And so it's the anterior or ventral end of the hippocampus that is critical for sort of representing and utilizing context. That's the claim. Well, about 10 years ago, and here's yet another collaboration with Morris, is Jordan in the room? I don't know if Jordan's in Toronto, but I don't know if he's in the room. I can't see. So a postdoc working with Morris and he was a graduate student, even better. He was a graduate student working with Morris and a colleague of mine from Norway, Halvard Evansborn, wrote a review paper in which we don't pay attention to all of this stuff here. Just look at the colors. What this shows you is that the anterior hippocampus is by and large talking to a very different set of structures than the posterior hippocampus. Right? There's a little bit of overlap in terms of the amygdala, but mostly they're talking to different networks. They're doing different things. So now there's two important considerations to think about in thinking about sort of this long axis. One of them is that theta activity in the hippocampus moves pretty much in a wave-like fashion, but only in one direction, from dorsal to ventral. Right? Neurons in the ventral end of the hippocampus have relatively limited interconnections going back the other way. So it's a one-way street, more or less. And I'm going to stick something in here that wasn't on the slide. I know a lot of you are interested in ripples, in hippocampal ripples, how important they are, reactivation. They are mostly generated in the dorsal hippocampus, not in the ventral. The vast majority of the ripples that actually have an impact on what's going on are generated in the dorsal or posterior hippocampus. That's for future thinking for those of you who care about that. Anyway, there's a unidirectional flow of activity that looks like it's going from the small grain to the big grain. Drew Maurer and I used this in a TICS paper a few years ago to propose kind of a notion that the hippocampus was absolutely crucial for establishing the continuity of context, sort of that I'm still in the same context. So here's roughly the story. The dorsal hippocampus, the posterior hippocampus, constantly detects local change because of the small grain, right? It's picking up all kinds of small change. And that could reflect just local sensory drift. As you move ventrally along the axis, this grain coarsens, and this local change is smoothed over, unless it's extensive. Right? And this arrangement of kind of small change being smoothed over and only big changes actually propagating down the axis enables the detection of distinctive boundaries. This is what we think, or how we think the hippocampus does boundary detection. And this allows the hippocampus, this arrangement would allow the hippocampus to monitor and address an absolutely critical question for the organism. Given the environmental change, however much there was, am I still in the same context? That's a critical question, basically. And again, because when you shift from one context to another, lots of things happen. Think about restaurant behavior that you tell your kids, right? What you say at home and do at home, you can't do in the restaurant. So contexts matter. Am I still in the same context? Yes or no? So how can this work? Here's an idea about how it could work. And it depends upon mismatch, which I'm using instead of prediction error, but let's assume I'm saying the same thing here. If a little bit of change is detected at the dorsal end, it never gets past the dorsal hippocampus. It doesn't get communicated down the line. A little bit more change, some of it makes it to the next level. And so on, a little bit more change, some of it makes it to the next level. Finally, with enough change, these orange things are kind of the mismatch elements, the prediction error elements. With a sufficient amount of change, suddenly the system decides, I'm at a boundary. And that then generates a kind of a system reset signal that tells the animal, you're not where you thought you were, stop thinking you're in that place, and start thinking something else, right? And we think, well, just another sidebar, of course, there's plenty of evidence now that the hippocampus is involved in boundary detection, both in rats and in humans. I won't dwell on that. So, interim summary number 2. The hippocampus creates these spatial context representations that reflect the specifics of a particular place, what's in it, what happens. They allow animals to re-identify environments and to accurately map across time, to aggregate information from multiple visits. There's an increasing grain of the representation as you move along this long axis of the hippocampus. And the shifting scale allows the system to ignore minor changes and accurately identify and update environments. I'll get to that later. But major changes indicate boundaries, either in space or time, and these generate sufficient prediction error and a system reset. And we think this is probably mediated by the locus ceruleus. And so we're now working on a kind of a model of how the locus ceruleus aggregates prediction error and then influences the memory dynamics in the hippocampus, such in a way that says, block the context representation you initially activated, that was wrong, create a new one, or update the one that you initially activated, either of those outcomes. So the possibility of this kind of a system reset is particularly important with regard to memory dynamics. So this we've heard a fair amount about at this meeting already. An organism is constantly faced with a choice, or its brain is, so to put, between integrating new information with existing knowledge or creating a new memory, a separate memory by itself, because it doesn't fit into what you previously thought it was, right? And the study of so-called memory reconsolidation focuses on this issue. So some years ago, Morris Moscovich and I proposed the multiple trace theory, which challenged the standard model of memory consolidation. Now, Morris didn't show any pictures of me, so I thought I'd take advantage of him. So, amongst other things, MTT suggested that reactivated memories, even remote ones, can be altered through a process that's come to be called memory reconsolidation. Now I'm going to reconsolidate Morris to something closer to what he looks like now and tell you the story of what this led to. So many of you, I'm sure you all know the traditional consolidation story. It's in all the textbooks. It's as wrong as the separation between perception and memory is wrong. It's one of those things that's in the textbooks that doesn't match reality. But here it was. This was what I learned, and this is what I taught. Something happens, you encode it, you consolidate it, and you got a fixed memory. End of story. Now, of course, we know that's not the end of the story. We now know that if you reactivate a so-called fixed memory, it gets unfixed in some way. And if you, at that moment, if you happen to be working with rats and you can inject a protein synthesis inhibitor into the amygdala, because this was done with fear conditioning, you can basically delete that memory or erase it, or apparently can do that. If you, at that moment when the memory is unfixed, when it's labile. This was the Nadel and Ledoux study in 2002, but it actually followed on an earlier study by Susan Serra, the same one who talked about system reset in the locus coeruleus, by Susan Serra and a student whose name is not pronounceable, Przybyłowski or something, a very long Polish name. In 1997, they essentially showed the same thing in a maze study that Nadel and Ledoux showed some years later. It's worth looking up. So when we started to think about this, when you unfix a memory, you don't want it to be the case that every time you think about a memory, that it's kind of going to get knocked out. That doesn't make a lot of sense. That doesn't make evolutionary sense. So there must be something else going on here, and it's about updating your memory, right? It's about gathering some new information in a situation that you recognize, that you identify as similar to the one you've been in. You unfix the memory because you've activated it, you've retrieved it, and now if there's something that's been slightly changed, you went away and somebody moved the furniture in your living room while you were gone, you can remap easily. So that's this updating function seems to be the purpose for this unfixing of retrieved memories. So we decided we would study this experimentally. We, in this case, being a postdoc, Alma Hupback, who's now at Lehigh University, and a graduate student, Oliver Hart, who is now at McGill, and a colleague at Arizona, Rebecca Gomez. So we did this experiment where it's kind of like a list learning experiment, and the participants learned about a bunch of objects. These objects were pulled out of a blue basket one at a time, and they were, this is a comb, and just remember all these objects. And they reached a criterion, I think 16 out of 20, we had them go to criterion, or 4 trials, but they all reached criterion before that. Then they went away, and 2 days later, they came back on a Wednesday, and they were split up into 3 groups. I'm only going to tell you about the 2 critical groups, a reminder group and a no reminder group. The reminder group was brought back to the same context with the same experimenter, and they were asked a kind of a leading question, like, what did you do here the other day? And they said, well, I learned about a bunch of objects. Okay, that's enough. We didn't ask them to retrieve them. The no reminder group was brought back to a different context with a different experimenter, and they were given no such question, right? And then 2 days later, they were brought back to the original context, and they were asked to recall the first set, right? So what we expected was that the participants who'd been reminded would somehow add some of the items from the second list to this destabilized first list. And that when we asked them to give us the first list back, they would give us some of those second list items, and that this would not happen in the non-reminder group. And that's, of course, obviously what happened, otherwise I wouldn't be telling you about it. So these are the intrusions of second list items into the first list when we asked them to recall. This is in the reminder group, this is in the no reminder group. So we have a pretty strong effect. But we have 3 kind of triggering conditions here: the context, the experimenter, and the question. In a follow-on experiment, we did many follow-on experiments, but in the one I want to tell you about, we separated these 3 out, so they only got one of them. They either got the same context, or they got the same experimenter, or they got the question without the other things. And what you can see very clearly is that it's about context. The context is the best retrieval device for destabilizing their memory of that first list, and that opens up the possibility of these memory intrusions, that so-called memory intrusions. So context can play an important role in determining whether a memory is reactivated and updated. And by reactivating old memories, context enables updating that, again, can effectively bridge the passage of time. So this memory updating phenomenon kind of parallels contingency learning. It involves aggregating information over multiple learning occasions to more accurately model the environment. And that's what's adaptive. So the takeaway, as I'm getting close to the end here, the hippocampus, by creating context representations, enables organisms to bridge time and space. This affects both ongoing behavior and the ways in which we create and update memory so that we can behave more adaptively in the future. Now I'll just take a step back and go back to the goldfish. So Many of you know, of course, that the hippocampus, or something like the hippocampus, goes back in evolution 350 million years to the first jawed fish. Not quite clear why having jaws would lead to having a hippocampus or something like it, but that part is yet to be determined. Anyway, the equivalent of the hippocampus in the goldfish and other things like lizards and turtles on the pallium is, according to this paper that came out, I think, in 2017 or something like that, from a wonderful group that does comparative neuropsychology in Seville that I visited maybe 20 years ago, a fabulous group. They've just shown now that the hippocampus in goldfish is necessary to associate temporally discontiguous events, the same claim that I'm making from the rat in the human literature. All right, now you might think, why do I frame this as a story? Well, sometimes I've given this talk in a way that I ended with a description of some recent research done with a former student, now collaborator, who's at UC Irvine, on what actually makes a story or a narrative credible, right? But if you want to hear about that part of it, you have to, like me, get up tomorrow morning early and show up at the poster session where I'm stuck somewhere near the men's room at D159, and I can tell you about sort of what we have learned about, what makes a story credible and what makes it not so credible. To close with George Miller again, just want to point out that, look at the things I've bolded. He was talking about context. This is in a 1950 paper, and it was clear that in that 1960 book, the mechanisms and the models they were talking about depended on something they were calling an image. And that image, this is a review by Peter Milner, by the way, the person whose course I was TAing when Morris was in it. This was actually a cognitive map. So George Miller was already thinking about context and cognitive maps way before I came on the scene. I met him once. He's a wonderful guy, and again, it really was an honor to receive this award. And with that, I will thank my collaborators, and I hope you enjoyed my story. Thank you. Now for the coda. So we all know about Endel Tolving, of course, and there's going to be a symposium in his honor, and then it's an event on Tuesday night. And of course, Endel was well aware of the fact that we can do pretty weird things with space and time, and that time's arrow is bent in a loop when we think about the past, and it's not every organism can do that, but we can bend time by using this system, I presume. Anyway, thank you. I'm told there's time for a question or two if you want, but I'm also okay if there are no questions. The poster session is starting very soon.